So I think we're good. I see a lot of people who have registered, have joined us. So again, hello everybody. Good afternoon, good morning. We have different time zones. It's a pleasure to have you with us today and to have Dr. Tapinos giving us this interesting talk on RNA epitranscriptome in brain tumors. So just before I give the floor to Dr. Tapinos, I will just give a brief introduction of his work. Let's start. So Nikos is an associate professor and an endowed chair of neuroscience and neurosurgery at Brown University. His work focuses on the intersection of RNA biology and epigenetics in cancer. He completed his MD and PhD at the Medical School of Athens in Greece, where he published seminal papers on his discoveries of a new mutation in P53, Sjogren's syndrome, and the prevalence of Coxsackie virus infection as a cause for autoimmunity Sjogren's. He then joined Rockefeller University as a postdoctoral fellow, where he published papers in Science, Nature, and PNAS on molecular mechanisms of peripheral nerve injury response. Nikos is a part of the Human RNA Project group at the Academies of Science, which aims in writing a white paper addressed to the US Congress about the need to map and sequence all modifications of human RNA. This project is expected to be double in scope compared to the Human Genome Project. In addition, Nikos is a founding member of a new RNA center at Brown University. At his lab, he developed the Epicodem RNA discovery platform that combines AI-based utilization of the largest 3D structure library in the world, next-generation sequence-based mapping of RNA modifications at the single nucleotide level, and chromatin landscape mapping. In 2023, Nikos founded Tapinos Therapeutics, which is utilizing the Epicodem RNA discovery platform, leading to key discoveries regarding targeting the epitranscriptome for therapeutic purposes and the development of first-in-class RNA modifier molecules and epigenome editors. So as you hear, we're going to hear from him a lot of very interesting data and about new discoveries in brain tumor research. And just to note, whoever has a question, please note it down at the Q&A section, and we will be addressing that at the end of the talk. Thank you again for joining us. Thank you, Nikos, for being with us today, and the floor is yours.
Thank you, Maria, for the introduction and for the invitation. I'm very happy to be here and give this talk about our work on the RNA epitranscriptome in brain tumors. And let me start with a brief introduction about brain tumors. And as you see on the far left of this slide, this is a typical MRI of a patient with glioblastoma that comes to the hospital. And when they do the MRI, they see this lesion in the brain. So the typical course of action is to do surgical removal of this lesion. And you see here on the MRI that post-surgery you have this edema forming because of the surgical procedure. A month later, the patients usually start a combination of chemotherapy and radiotherapy. And in the MRI, you just see nothing, basically just an empty hole where the tumor is, which is a very good indication that it was removed completely. 3 months later, there's maybe a little bit of a flare, but most probably edema as well, so nothing really serious. But 7 months later, in this case, you have a new tumor at this site and maybe even at another side of the brain. And that's a usual course. The 5-year survival rate, unfortunately, for glioblastoma still is less than 6%. And the prevalence of glioblastoma in the United States is around 13,000 people per year. The mean survival is not more than 18 to 20 months, I would say, right now. And if it's starting to see that if you do a comparison of 5-year survival rates between different cancers, for example, pediatric leukemia or breast cancer, where you see 9 out of 10 patients, if there is an early diagnosis, they can actually live more than 5 years. When you talk about glioblastoma, you are actually less than 2 people every 10 people, and deep G, which is the pediatric glioblastoma, is basically none. And here is some very bad, statistics about the pediatric glioblastoma. It's a very rare disease. There are around 300 cases in the United States per year, but more than almost 100% of these children die within 2 years of the diagnosis, and the median survival is 9 months. The very sad statistic is that there is no improvement in survival rate of children with deep G or any therapy, specific therapy that has been developed in the last 40 years. So going to the why these diseases are so difficult to treat and so difficult to even understand, what we know because of work of many, many labs and many people around the world have actually proved that one major component of glioblastoma is a population of cells which are called cancer stem cells, or you will hear me calling them glioma stem cells. And why these are important is that you will see here, this is data from my lab. So this is a 3D light sheet microscopy of a human glioblastoma in a mouse brain, and you will see the tumor mass. If you do a regular resolution image, like a 3D stack image, you will see and reconstruct the tumor. You will see that the tumor actually delineates very nicely. So a neurosurgeon can basically easily remove this tumor in a brain of a patient. This is obviously a mouse, but we're saying that this can be removed. Now, if you go to actually the same tumor, if you use high resolution and increase the resolution, increase the depth of imaging, what you see is that this is the reality. The reality is that there are thousands of cells which have already invaded the parenchyma of the brain, and most of these cells retain this stem cell ability, which means that they can form recurrent tumors. And that is one of the major problems why we cannot treat this disease so far. So, and we and others, like years ago, we put together this concept of stem cell plasticity in glioblastoma. You see here a beautiful image that John Zepecki in my lab did several years ago. This is a human glioblastoma which is growing within a network of cortical neurons, like in blue. And as these cells invade the neuronal network, actually they express nestin, they express Ki67 because they proliferate, but they also expressed GFAP, which is a differentiation marker. And this is actually very weird how a cell can express both stem differentiation markers. So with this and more other work that we have published, we actually put together this concept that the cancer stem cell in brain tumors actually is in a plastic state, which can differentiate and de-differentiate and become a poised cancer cell. The differentiation or de-differentiation cues come from the microenvironment of the cancer, like the autocrine signal, paracrine signals, hypoxia plays a role, the rigidity of the extracellular matrix play a role. But within the cell, as the cell actually accepts all these extracellular cues, there are different mechanisms of DNA and histone modifications, non-coding RNAs, alterations in chromatin, transcription factors. And lately, we know that RNA modifications and in particular M6A play a major role in regulating this cancer stem cell plasticity. So we decided several years ago to focus on RNA modifications among all the other stuff that we're doing in the lab. And that will be now a segue into the major part of my talk about the role of the M6A RNA epitranscriptome in glioblastoma. So before going into the details of the data, what is the RNA epitranscriptome? So what we know now is that if you think that you have a DNA, this is, I have to say it from now, so it's clear that this has nothing to do and does not depend on DNA mutations. So even if there is a mutated DNA or a gene, or if it's non-mutated, as it is transcribed to RNA and before RNA gives rise to proteins, there is a whole mechanism in the eukaryotic cells, which is called the epitranscriptomic machinery, which either instills like modifications on M6A on this adenosine, and we know now that there are enzyme writers, that the METTL3, METTL14, WTAP proteins that actually put modifications. There is two proteins so far identified, which are the erasers of M6A modifications, and this is ALK-BH5 and FTO. And then depending on how is the methylation pattern of the transcript, there are reader proteins, which is the YTDC1, the YTHDF1, and YTHDF2, which bind to the methylated RNA based on the ratio of methylation that happens on this transcript, and either take the transcript to translation to the ribosome or to degradation, as you see here, through an endoribonuclease pathway with a C-NOT and non-mediated decay of the mRNA, depending on the level of methylation per transcript. So this is a very interesting mechanism, and it's a regulatory mechanism that actually gives a message to the cell that the transcript is fit for translation or unfit for translation. It has to be degraded. So the last, I would say, 6 or 8 years, people have started working on different aspects of the epitranscriptome in glioblastoma. There are papers on METTL3 and METTL14, the writers, the papers, especially from Jeremy Ritz's lab and Chuan Hian's lab, that about the YTHDF1 and 2, the readers in glioblastoma, and also the ALK-BH5. But the FTO, the demethylase FTO, has not been published for the role of FTO in glioblastoma, was not published for several years until we published it like some years ago, that FTO has a major role in regulating translation in human patient-derived glioma stem cells. And what we saw is that FTO actually regulates the levels of nascent translation by forming a complex with Argonaute-1 and ILF3. And this is microRNAs also participate in this complex. And this complex regulates how transcripts are translated in cancer stem cells, in glioma stem cells. And not only this, what we did on this work, we actually did a combination of M6A RNA-seq and ribo-seq. And we show that consistently, and this is the data that I'm showing here from 3 individual patients that came to Rhode Island Hospital. And we isolated the glioma stem cells from these patients and did this work. And we saw consistently that the translation efficiency of in glioma stem cells actually increased when the M6A, the level of M6A is decreased. So the more M6A you are losing, as you see in these 3 patients, the more efficient translation happens, which you can imagine the opposite also is true. The more methylation you have, and you see it here, then the less efficient translation is happening. So we wanted to see if this is, what is the role of FTO on this mechanism? Is this specific to FTO, or is FTO playing a role on this mechanism at all? So we did a knockdown of FTO, and we did this very, in my opinion, very elegant study at the single molecule level, where we, by knocking out FTO in the cells, in these glioma stem cells, we actually did a pulse chase experiment. And we saw here, this is a one protein, but one transcript, but obviously this is true for other transcripts as well. This is CLIP3, and it's a tumor suppressor transcript. And you see in the control cells, these dots, these yellow dots that you see in the cells are actually single molecules of nascent CLIP3 going to the ribosome to be translated. When you inhibit FTO, which basically means you hypermethylate the transcript of CLIP3, so there is no translation. So you inhibit very significantly the translation of CLIP3 and other transcripts which are hypermethylated. So this was a very, very interesting and very important hint to us when we finished this study, that FTO could be an interesting and important, even therapeutic target. And the concept that we put out, if just I'll put it out there from at this stage of the talk, is that imagine that if FTO can do that, that means that if you can manipulate the levels of FTO and you can manipulate the levels of RNA methylation in these glioma stem cells, then you can trick the cancer cell to think that transcripts that are being produced are not fit for translation. They will go to degradation, and that will be detrimental to the cancer cell. So the first thing that we did in order to see if that could be targeted as for potential therapy, we actually tested the expression levels of FTO in glioma stem cells. This is like glioma stem cells from 5 patients, as you see, and then we tested it with normal human neural stem cells, normal human astrocytes, and total normal brain lysates. And in all instances, the glioma stem cells have higher expression of FTO, which seems that they're depending on this demethylation that FTO is doing. And it's very interesting that we think now we are very confident that FTO is an excellent therapeutic target because other people have shown the role of FTO as tumor promoting in other cancers as well, not only brain cancer, for example, colon cancer, non-small cell lung cancer, ovarian cancer, pancreatic cancer. And the interesting thing is that FTO has way lower expression in healthy tissue relative to cancer. I will show you some data later on. And another very interesting thing that you have to keep in mind that FTO is a very specific demethylase, so actually has the ability to recognize secondary structure of RNA. So it can recognize the stem loop structures of RNA and not the linear RNA strand. So that gives FTO a very important specificity for demethylation. And why M6A is important? It's important because it's the most abundant RNA modification in eukaryotic mRNA. It controls levels of mRNA. Basically, it regulates if the mRNA will go to degradation eventually or to translation. It controls even or regulates alternative splicing. And it's also the most quantifiable RNA modification. So these all telling us that FTO is a good therapeutic target and we should proceed forward. And how did we end up forming a potential drug to target FTO? So as Maria said in the beginning, we have, with collaborators, we have established and we have access to this extremely large library of compound library that holds 1.2 billion compounds. And the three-dimensional structure of FTO has been resolved. So we took the eukaryotic, the human FTO three-dimensional structure, and using artificial intelligence powered in silico binding prediction, we actually compared the enzymatic pocket of FTO with this 1.2 billion compound library. And the initial filtering gave us 60,000 compounds that could potentially bind efficiently to FTO. Then from there, we went into a lot of refinement and filtering with selecting the high potency compounds for binding affinity, the compounds that could potentially be synthesized, or feasibility of synthesis. And also we added another filter to identify compounds that potentially in silico could cross the blood-brain barrier. So this all filtering actually reduced the library significantly down to 7 candidates, which we synthesized all of them. We have them in the lab. So they're all FTO inhibitors, and we tested with enzymatic assays targeting the cell-free demethylase activity of FTO. We identified the lead compound, which we call TNO1, and this is actually, the selection is based on the nanomolar potency of this towards the demethylase activity of FTO. And to show some data about the drug, it's a small molecule inhibitor. So if you incubate the small molecule with human FTO, it stabilizes, it binds specifically to FTO. It provides thermal stabilization of the protein, and this is concentration dependent. It actually very significantly stabilizes FTO even at a very low levels at 500 nanomolar, and this shows specificity of binding. But most importantly, if you take this inhibitor and you treat in very low concentrations, if you treat either on the left, you see here a dot plot, an RNA dot plot. If you treat the colon cancer cells, or even the GB2 is a glioma stem cell line from a patient, and you treat these cells with this inhibitor, you increase total levels of M6A, as you see in these RNA dot plots. But of course, RNA dot plot is a semi-quantitative technique. So we went on and we performed RNA HPLC on the glioma stem cells, treated or non-treated with our inhibitor. And what we're showing is that we can increase by 51% the level of M6A methylation globally in this cancer stem cells, patient-derived cancer stem cells. So the inhibitor does what it's supposed to do. Now, as I said before, the concept was that if we hypermethylate transcripts, then that what we believe is going to happen is that we will affect transcription and eventually translation. So in order to start answering this question, if this is happening, we did this combination of RNA-seq and M6A MERIP sequencing, and we found that indeed the hypermethylated transcripts are actually having inhibited transcription. Obviously, there are some which are also increasing transcription, as you would expect. This is the here you see the distribution of hypermethylated transcripts and after treatment with our inhibitor, and you see that the ones that are hypermethylated belong to very important functional classification categories like NOTCH, ROD, GTPases, and F-mediated invasion pathways. So this tells us that the hypermethylation really affects transcripts with a major function in glioma stem cells. But again, going back to our ability to use and the EpiCode RNA platform that we have developed, ability to understand at a single nucleotide resolution what is happening on methylation. I'm showing you some data of exactly this. This is mapping of every adenosine on the genome of these glioma stem cells using single nucleotide mapping of methylation after treatment, after inhibition of FTO. And what you see here, what is happening, the distribution of M6As that are hypermethylated. And if you, for example, you want to choose your transcript of interest, here is the Notch family of transcripts. So it's not only Notch 1, but all the other transcripts that actually work with Notch. You can actually go to each one of these transcripts, and the dots that you see here on the right are actually adenosines, either in the CDS or the 5-UTR or the 3-UTR of these transcripts that are hypermethylated after inhibition of FTO. So you can really map and understand what you are changing per transcript. And again, the question is this FTO specific? How do we prove specificity? Then we took our MERIP-seq on this from these GSCs and compared it with a CLIP-seq on FTO. And what we saw here is like almost perfect correlation of what is happening on the hypermethylated adenosines and the FTO binding. And if you do an FTO, and this happens both on mRNA and long non-coding RNA. And if you actually identify the motif where FTO binds, and this is the motif, this is the most significant motif where hypermethylation happens. So the effects that we're seeing globally are mostly because we're affecting FTO binding and FTO demethylation. So, but what is functionally, what does all this mean? So one of the most important aspects of cancer stem cells is this ability of the cancer stem cell to self-renew, which means that if you take a single cell, it can actually form a sphere and a tumor by itself. It's a clonal expansion. So we did these tumor sphere formation assays from 2 patients, and now we have done it with another 5 patients with gliomas from glioma stem cells. And very low, again, because this is cellular treatment with 500 nanomolar of the inhibitor, it actually inhibits significantly the ability of cancer stem cell to self-renew. And this is one of the hallmarks of cancer stem cells, is the ability to form clones of themselves. But the other hallmark, especially because we're talking about glioblastoma, is the inherent ability of these cells to invade and to migrate within the tumor parenchyma. And I will play here 2 videos just to show, to actually stress this point. The left video that you see already here, it's actually a video of human glioma stem cells transfected with GFP, and we put them on nanofibers, and without really a gradient, without really growth factors attracting them, the cells start migrating by themselves. This is an inherent ability to just move. And if you establish a co-culture of glioma stem cells, as you see this here again with GFP and axons, which we have done it in the lab, and you put the glioma stem cells on the axons, then they are very happy about it. They invade the whole neuronal network, and they actually even eventually destroy the neuronal network. So this is what these glioblastoma cancer stem cells do. They invade the parenchyma and they migrate without even having a growth factor Q. So it will be very important if we inhibit transcripts or hypermethylate transcripts and eventually make these cells to stop this invasion capability of these cells. And this is exactly what we're showing here. So we took from 2 patients glioma stem cells, and you see how, as they grow, we took, this is a stationary picture on the left on the control, when the glioma stem cells formed the sphere and then they invaded the parenchyma to the end of the slide that you see here. But when they are treated with our drug, here this particular experiment is with 1 micromolar this drug. So, and 10 days later, you see that we're significantly inhibiting invasion of these cells. So our treatment affects both stemness and invasion. So that means that we are targeting the 2 most important abilities of the cancer stem cells to invade. But as I said in the initial work, when I was showing that we designed the drug to be able to cross the BBB. So we tested that. We worked with Charles River, and we tested an in vitro model of epithelial crossing with the MDCK cells. And the FDA permeability cutoff for BBB is less than 3 times 10 to the minus 6. We are actually at 15 times 10 to the minus 6, and you see it here. Our drug efficiently crosses the BBB, and you see here the negative controls do not, and this is the positive control. So the design is actually did what it's supposed to do. And when you test the drug on cytotoxicity of normal cells, if you recall what I showed you about self-renewal on tumor sphere formation, we use 500 nanomolar. In order to have an effect in human astrocytes, you have to increase up to 75 micromolar to go to IC50. So it's pretty safe. There is this window of much lower FTO expression in healthy cells versus cancer, which gives us this ability to use this inhibitor quite safely. And we also did the HERC potassium channel assay for cardiotoxicity, because a lot of drugs that are small molecule drugs, they do have cardiotoxicity. And we did this work again with Charles River, and we didn't see any cardiotoxicity from this drug. So it has a lot of very how can I say it checks a lot of boxes to be further developed as a potential drug. So let's see some in vivo data, because all this is in vitro. So in order to see if it has an effect in vivo, we actually used patient-derived glioma stem cells. We did the PDX model on brains of immunocompromised mice, and you see here the randomization day. And then 28 days, we actually in this model, and I will show you systemic delivery later on. So in this model, we used an LZ pump, which gives the drug directly in the brain. So it's an intracranial delivery. And what we see in a very low dose for in vivo, 1 milligram per kilogram per day, the pump holds enough drug for 21 days. So 28 days post-pump, this is what we saw. We saw that 2 mice have complete elimination of the glioblastoma, and the other 3 were actually very low comparing to control. So we decided at this point to leave the mice there. They were not getting any more drug, to see if the tumor will come back, as it usually does, because it's glioblastoma. So to our surprise, this is 60 days. Not only the tumor didn't come back, and don't forget from day 28 until now, we're not giving any more drug. Not only the tumor didn't come back, but now 4 mice out of 5 have no evidence of tumor. So this seemed that, we're completely eliminated the tumors, which is even scary to say when you're talking about human glioblastoma. But then we said, okay, let's go now a little bit, crazy and wait another 60 days. So we go to 120 days. And 120 days, all the control cells are dead because of the tumors. And the mice are not creating new tumors. Maybe some signal on this mouse, but there's no tumors. And this is, it has never been seen before in human glioblastoma. It's very exciting data. So, but this is intracranial delivery. And we wanted to see what happens with systemic delivery. So we collaborated with Certis Oncology in California. And so they did these orthotopic glioblastomas from patients in Mayo Clinic. And what you see here on the left, it's actually the vehicle controlled. So we're testing with MRIs here, the mice. So in day 0, we have equal small tumors in the mice, and this is the treatment. Oh, I forgot to say that this is oral treatment with gavage, which again, giving a drug orally for human glioblastoma, it has never really been shown efficacy before. In day 21, all the control mice were dead with the tumors extending, occupying the whole hemisphere, as you see in this MRI, even extending to the contralateral hemisphere. It's a disaster. It's very big tumors, 107 cubic millimeters. This is what happens with the drug. So the tumors that grew actually are half the size of the other tumors. And most importantly, the shape of the tumors actually is very regular, which reminds, as we say in the lab, meningiomas, not really glioblastomas. So which is very interesting, which means that probably invasion is not happening. But there were also tumors and some mice that the tumors never grew. So this is from oral delivery. And if you look at the systemic toxicity, there's no toxicity. And if you do Kaplan-Meier, obviously the tumor, all the mice were dead at 20 days, the control. The others were perfectly fine and alive. So we have very high significance on the Kaplan-Meier. Then we also in the lab repeated the systemic delivery by giving it intraperitoneally. And we see again the same, very significant inhibition of tumor growth, significant effect on survival, and tremendous difference on growth of human glioblastomas. This is subcutaneous and IP delivery. So as we were seeing this data, we said, okay, FTO is overexpressed in other tumors as well. And we got this colon cancer samples from healthy on the left and colon cancer on the right. This is healthy colon, the expression of FTO on the left, and in the colon cancer, I don't even need to tell you that this is super overexpressed in colon cancer. So that could be a very good indication, and we wanted to try. So we took colon cancer cells in the lab. We performed in vivo experiments, again, with systemic delivery of the drug as a monotherapy, without chemotherapy, without anything else. And we still see very significant inhibition of tumor growth in these cells with human colon cancer subcutaneous flanks. So finally, and this is the latest very exciting results that are coming up from the pipeline in the lab. So one of the very interesting proteins that we show the transcript is hypermethylated, and then we did the Western blots, and we're showing here almost complete knockdown of this protein is this the immune checkpoint, CD276. This is a cancer-specific immune checkpoint, which points to a direction that by inhibiting these important immune regulatory molecules, we may increase immune activation. Then at the same time, and as I introduced in the beginning, M6A methylation also regulates alternative splicing. So we analyzed alternative splicing on glioma stem cells based on our RNA-seq data, and we found that really if our drug has a tremendous effect on several aspects of alternative splicing. It changes completely exon retention, intron splicing. So really the alternative splicing is significantly altered after hypermethylation that is happening. And what does this alternative splicing mean? It means that there is a possibility that if you do this in cancer cells, you are actually making new antigens that the tumor cells may recognize and attack. And in order to test this, we did this human PDX model, organoid model of non-small cell lung cancer. This is with collaboration with CyPre, which is a subsidiary of Charles River. So we did this human-derived non-small cell lung cancer tumor organoid with or without PBMCs and with or without our drug. And what we're showing here, that we're really increasing the cytokine production of GM-CSF and CD40 ligand, which tells us that there is a antigen-specific tumor T-cell expansion in these cells, and the T-cells were attacking the tumors. So this is a very interesting spin, that how we can use an M6A modifier molecule to change RNA modification levels in cancer cells, and this gives basically priming the tumor for immune attack. This is a completely new concept. And as to conclude my seminar today, so I want to tell you exactly what I show you today, is that we have DNA is transcribed to RNA. There are M6A writers that actually modified M6A on these transcripts before going to translation, and erasers that remove them. Our drug blocks the FTO eraser, so you actually keep the transcripts hypermethylated. After this hypermethylation, several things are happening in the cancer cell. One is transcript degradation. And what is the result of this? You see it here in a very beautiful tumor sphere from a patient with glioblastoma that it forms. This is a tumor spheroid. If it's treated with our drug, this cannot happen anymore. So they're losing the cancer stemness ability. But also through the degradation, we're affecting immune checkpoint molecules. And because of alterations in alternative splicing, we produce new antigens. And this is new data that came to the lab, that we're doing in the lab. What we're showing here in this very short video, you see a human lung cancer cell and then T cells that are actually co-culturing together, and there's no really interest of the T cells for the cancer cell. So when adding T cell, our drug, and we do a live imaging of this for 3 days. So this is what is happening. The T cells are activated, attack, and completely kill the cancer cells. So this is a very exciting angle for us, and we're going to pursue this further. So where we are now and where we're going, as Maria mentioned, we have incorporated a startup company to take this molecule to optimize it further, because yes, it does incredible stuff in the lab, but that doesn't mean that the same exact molecule can go into humans. So we are in the phase of lead optimization of this molecule, and then eventually GMP manufacturing and toxicology studies, with the hope that soon, whatever soon means, we can be on an IND and clinical trials for patients with not only glioblastoma, but glioblastoma, colon cancer, non-small cell lung cancer, and even other tumors. So at this stage, I want to thank people in my lab, especially David, John, and Laura. They are instrumental in this work, and other members of the lab, obviously, are my collaborators and the funding mechanisms. And again, thank you so much for hosting me today, and I'm happy to answer questions if you have.
So thank you, Nico, for the excellent talk. For some reason, my video cannot open. So I must say, the results that you just presented are mind-blowing, and especially when we're talking about glioblastoma. That's amazing. So I'm just gonna take the advantage of the host and ask like the first question, starting backwards. So the last video that you showed about with the T cells, that they were not interested in the tumor cells, and then when you added the drug, they completely destroyed them. So do you have any idea what's the exact signal that changes? Because if you think about it in the context of the tumor microenvironment, there's a lot of things happening. So and especially in the brain. So do we know if this is something of a paracrine signal from the cancer cells that signal to the T cells, or is it on the T cells that's happening, or is it both ways? I know it's complicated, the question, but
yeah, so that's a great question. It's not on the T cells. We have repeated this experiment. So we took T cells, human T cells and PBMCs alone without the tumor cells, and we treated them with a drug. So there's no effect on the T cells at all. And that's not really surprising. The T cells do not express FTO, is almost nothing. So we're not targeting anything on the T cells. So going back to your, of course, you know, the in vitro experiments and this, they are very simplistic, right? So you take the T cells and you have the cancer cell. That is very far away from reality and from the tumor microenvironment, which I agree with you 100%. But that also gives us the ability to start thinking how this could work. And we have several hypotheses that we're going after. One hypothesis is that based on alterations on the cancer cells on alternative splicing, there is probably the cancer cell can present on the surface new antigens that the T cells actually recognize, and you have a clonal expansion of an antigen reacting T cell. That is a possibility. Another possibility that we're looking is that because of the degradation, you know, hypermethylation, degradation of transcripts, there are some transcripts which seem to be very interesting on the glucose metabolism of T cells.
Okay.
So it could be that we're actually affecting this, and there's more now glucose available in the microenvironment to activate T cell responses.
Mm-hmm.
So that is another possibility that we're looking. There is also this immune checkpoint inhibition, right, that we saw the CD276. That could be also an effect. So it's a combination. I cannot really tell you it's one mechanism right now. But the fact that we are seeing this activation of T cells, it's very, very exciting. And it gives us this ability to say that this can actually affect more tumors than not only glioblastoma.
Okay, so we have two questions.
Yes.
First one is from Dr. Antonis Kirmisis, who is a professor at the University of Cyprus, and he does a lot of work on epigenetics. So Antonis is asking, is this small very exciting work, is this small molecule specific to FTO, or does it inhibit other demethylases like ALK-BH5?
Yes, that's also an excellent question. We did this work, so it doesn't affect ALK-BH5 at all. I mean, you can go up to, I don't know, we went up to, I think, 800 micromolar or millimolar quantities, and there's no effect on ALK-BH5. We also tested other demethylases, DNA demethylases, and it doesn't affect. It was designed specifically for the hydrophobic pocket of FTO, and it's not shared with other demethylases. So that's why I think we have such specificity. So we are good for that. So, yeah.
Okay, so our next question is from Dr. Pani Trifilis, who has done a lot of work on translational research and drug discovery. So Pani is asking and saying very exciting and promising data. Congratulations. And the question is, do we understand what happens at the molecular level to allow persistence of the hypermethylated status even when the drug is removed?
Yes, so the drug, when we remove the drug, we don't expect the hypermethylation to remain. That we don't, and because there is this compensatory mechanism from an FTO will come back and ALK-BH5 will do some transcripts. So we don't believe that. But why we think this is important, still very important as a therapy, is because when you induce this hypermethylation, you are inducing a cascade of events. So, and I'll give you what I mean by that. What I mean by that is that if you saw in the initial in vivo studies where we gave the drug in very low doses, but for 20 days. So if you are blocking translation of cancer cells for very important molecules of cancer cells for 20 days, so there is no opportunity of the cancer cell to come back later on because it's already lost the ability to self-renew, to propagate, right? So, and that is why we kept the mice for 120 days after, because we are thinking, okay, they will come back. They always come back. It's glioblastoma, right? So
that was crazy.
Yes, that was crazy.
Yeah. I mean, mind-blowing. That's the only expression that comes to mind. So hopefully this will translate into the clinical level, and that will be a miracle for this thing.
Yeah.
It's crazy. So so I don't see any other questions. I'm just gonna jump in again. So again, going back to the to the microenvironment of the glioblastoma. We know, and there's a lot of data coming out now in the concept of cancer neuroscience, that we see that the glioblastoma cells, they kind of, they're their own, an individual brain, if you want to say, malignant brain growing into the brain. So have you done, or are you planning to do studies to look into what's happening on that level in the microenvironment, and what happens to the actual brain circuitry on the molecular cellular level, and then again the higher level is an extra target, but in the lower levels first?
Yes, yes. So yeah, that's a very good point. So yes, we are thinking about, we have, first of all, to answer your question, no, we haven't done these studies. So, but the the communication of glioblastoma cells within the brain with neurons, for example, and the synapses that are happening with neurons and axons and the glioblastoma cells, which is basically in my mind, it's a no-brainer that this is happening. It's in the brain, right?
Yeah,
so
exactly.
It has to happen. And these are glial cells. So
yeah.
how, for example, hypermethylation affects this communication? We don't know. Very interesting question, and we would love to study it in the future. Another thing that we're very interested to study, and we'll start now discussing, is that you have glioblastoma, and it grows in an environment, because of my bias on glial cells. What happens, for example, we threw this question out there. What happens to compact myelin of oligodendrocytes in the vicinity of glioblastoma? And how this affects it? Is there a repair mechanism that is happening? Are we blocking this? How does it work?
Yeah.
So there are a lot of questions that we don't know. But here, and these are very interesting questions for the basic level, but if you think translationally, right now, as you know, a person who comes with glioblastoma, the first thing that happens, they do a surgery, right?
Yeah.
And that changes everything. Then it's a completely different discussion about microenvironment after that.
Yeah, because you have no other option, obviously.
Yes.
I guess we're stuck with the models for now until we can translate.
Yeah, exactly. I mean, the dream, honestly, the dream is that, and I don't know if we are the ones, I hope we are, but I really hope that somebody else does it, even for these patients. If we can find something that we can shrink the tumor or stop the tumor, make it there, so you don't need to go immediately to remove it. You don't need to operate in the brain.
Yeah.
Brain is such an elegant organ. You don't go there and just removing stuff.
Yeah,
right?
exactly.
So if we can do that, that will be great.
Maybe a combination of the drug that you guys discovered with another one that actually limits the potential of the tumor to grow, like the tumor's organ, if you want to say the tumor's brain. So that would limit the communication, because we've seen that the glioblastomas actually control remote brain areas. Even if they're in one side of the brain, they can control other brain areas, which is, again, scary. I mean, the perspective of this tumor. So again, I want to take advantage. No other question is out there. So you mentioned at the beginning that these stem cells, the glioma stem cells, they have this dual status. So they actually express stem cell markers, but they also express GFAP. So do we know what are the steps that proceed to that happening? Because you, on the genomic level and even in the epigenetic level, you must have something preceding that to allow it. What's the landscape of the control elements on the DNA?
Yeah. And before answering the question, I would say something that I'm not a fan of markers for glioma stem cells. Because I don't think that markers tells us anything. These are a transient expression of molecules. The cell can actually change what it expresses. So, and how can a cell can do that, right? So if you do classic studies of chromatin structure, right? So you would expect that a cell that has actually decided to go to a differentiation trajectory that actually limits areas of chromatin, compacts the chromatin, and there are specific areas where are open and can do drive transcription. And this is the classic model of differentiation trajectory. So in cancer stem cells, and especially in glioma stem cells, that doesn't happen. So the chromatin remains in a constant open state. So, and this is another project that is running in the lab. So we did electron microscopy on patient-derived glioma stem cells, and you cannot really see heterochromatin in the nucleus.
Wow.
Which means the
crazy.
chromatin is in an open state constantly, right? And if you go back, I was very privileged, like many years ago, to be at a seminar at the Kaspari Auditorium at Rockefeller from Helen Blau from Stanford, where she actually put together, the title of her talk was a stem cell, is it an entity or a function?
Okay.
And she actually was actually putting it out there that she believed, and I believe 100% the same, that stem cell is a function of a cell based on what is happening around it.
Okay, yeah. Yeah, because you have reversion of like differentiated cells, like in cancer, back to a stem cell position. Now that you mentioned that, I mean, it comes to mind, like if you have open chromatin all the time, aren't these, isn't the DNA prone to damage and like breaking?
Yes, absolutely, it is. And there is a level of genomic instability in glioblastoma, which is at a very high level. Right. So if you do a chromosomal mapping of infusions of glioma stem cells, I mean, if I show you our data, it is a disaster. I mean, you cannot believe that this cell can be alive. It has like 4 times chromosomal arms. Chromosomes are missing. There's a huge level of genomic instability, but there is also a tremendous mechanism of maintaining the cell alive. So there is a lot of, as we discussed before we joined, there's a lot of helicases that play this role. They're actually maintaining the cell. And don't forget that glioblastoma is not a mutation-heavy tumor.
Yeah,
right?
Yeah.
So there are compensatory mechanisms that help the cell to do whatever it wants.
So it's an amazing survival machine at the end.
Yeah.
So we have another question from, let's see, Mrs. Gina Stavro. Congratulations, really encouraging data. I was wondering whether the EpiCode data is publicly available.
So not yet. We're finishing this paper, and when we submit the paper, we will put it publicly available, so it's open access. Yes.
Okay. Yeah, I have a lot of questions, but I want to, maybe we can continue it in another discussion. But really, the data you presented is mind-blowing, and I really do wish that it goes on to the clinical aspect. It would be like a miracle, I know, for many people that have been affected and are affected at this point from glioblastoma, and I guess it would open the gate for treating other types of cancer. So good luck with that. I mean, it was a great pleasure having you in our series, and thank you for accepting. And we'll be waiting with, a lot of excitement to see the next developments in your research.
Thank you so much.
Thank
you
you
for
very much,
inviting me and for everything. Thank you.
It was a pleasure. Have a lovely afternoon or morning for everybody. Thank you. Goodbye.
Thank you. Bye.
Bye-bye.